You think the dinosaurs ruled the Mesozoic? No, plants ruled it. One group of plants, specifically, which appeared in the middle of the Cretaceous period and in 50 million years conquered this planet so completely that today 85% of all terrestrial biodiversity exists only because they exist. They restructured the climate. They built the tropical rainforests. They fed the mammals, armed the birds, domesticated the insects. And ultimately, they created the conditions in which a human being could appear. Flowering plants do not decorate this planet. They are its operating system. Before we go any further, please subscribe to this channel. Your support is what makes this work possible, and it genuinely matters. Thank you. Now, let's talk about a revolution. Not a revolution measured in years or decades or even centuries. A revolution measured in tens of millions of years, slow by any human standard, but by the standards of deep time, explosive, geologically instantaneous. A revolution so thorough, so structural, so pervasive, that it did not merely change what lived on land. It changed what land could support. It changed the rules of the game entirely. The story we are about to tell begins in silence. In a green world without color, without fragrance, without fruit, a world that functioned, that breathed, that teemed with life, but a world operating on a much older, much simpler set of biological instructions. And then, in some warm, shallow, unremarkable body of water, in a place that left no monument, no marker, no obvious sign, something changed. Something small. Something that, if you had been standing there, you might have walked past without a second glance. One flower. The first flower. The beginning of everything that follows. To understand why that moment matters, why it deserves 90 minutes of your attention, we need to go back further, much further. We need to understand the world that existed before the flower. Because you cannot appreciate a revolution without first understanding what it overthrew. Imagine the Earth approximately 150 million years ago. We are in the Jurassic period, the famous age, the age of giants, the age that Hollywood has made iconic. And the world is, by any reasonable standard, extraordinary. The continents are in the early stages of separating from the great supercontinent Pangaea, though they still bear the memory of their former unity in their shapes and in the organisms that straddle their dividing shores. The climate is warm, warmer than today almost everywhere, with no permanent polar ice, with shallow seas pushing inland across continents, with vast coastal plains baking in subtropical heat. And the vegetation. The vegetation is something our eyes would recognize as green, as familiar in its broad outlines, but alien in its details. There are trees, yes, enormous trees, conifers that would dwarf a modern redwood, cycads, those strange palm-like plants with thick trunks and crowns of stiff, leathery fronds, covering hillsides in numbers we no longer see anywhere on Earth. Tree ferns rising 20, 30 meters into the humid air. Horsetails, ancient beyond measure, clustering along riverbanks and lake margins. Ginkgos, a genus so ancient and so conservative that the ginkgo trees planted in city parks today are essentially identical to the fossils from this era. It is a world of extraordinary biomass, extraordinary antiquity, extraordinary structural complexity. But here is what is missing: color. There is almost no color in this world beyond green and brown. No reds, no blues, no yellows, no purples. The reproductive structures of these plants, the cones, the spores, the pollen-bearing organs, are functional but not ornamental. They are not trying to attract anything. They are broadcasting, releasing clouds of pollen into the wind, hoping that statistics will do the work of precision, that out of millions of pollen grains drifting through the air, some fraction will land where they need to land. This is the gymnosperm strategy, and it works. It has worked for hundreds of millions of years. It will continue to work for certain groups right up to the present day. Pine trees still do it. Spruce trees still do it. The wind-pollinated grasses that dominate the grasslands of the modern world still do it. But it is a profoundly wasteful strategy. It requires the production of staggering quantities of pollen. It requires a certain density of the right plants in close proximity to each other. It is largely indifferent to the identity of the recipient, and it produces no fruit. This is perhaps the most consequential absence in the Jurassic landscape. No fruit means no reward. No reward means no motivated dispersers. The seeds of gymnosperms fall where they fall, straight down or carried a modest distance by the wind. There is no strawberry, no apple, no cherry, no fig. There is no transaction between plant and animal in which the plant offers something sweet, and the animal carries the plant's offspring somewhere far away. The animals of the Jurassic eat the vegetation, certainly. Sauropods consume enormous quantities of plant material. Their sheer body mass requires it. Smaller dinosaurs, insects, early mammals browse and forage through the green world. But the relationship between plant and animal is essentially one-directional. The plant provides energy. The animal takes it. There is no negotiation, no mutualism, no architecture of mutual benefit. Now hold that image in your mind. Hold the green, fragrant-free, color-free, fruit-free Jurassic world in your mind, because we are about to watch it change. The world has already been described. The climate was warm, the vegetation gymnosperm dominated, the continents still fragmenting. What has not been described is the specific place and the specific plant where everything changed. Into this world, this warm, humid, gymnosperm-dominated, endlessly green world, we need to place ourselves mentally. Not as observers with the luxury of knowing what comes next, as inhabitants, as organisms for whom this is simply the world as it is, as it has always been, as it presumably always will be. Stand at the edge of a shallow freshwater lake, somewhere in what will eventually become northeastern China. The water is warm and slightly turbid. The surrounding vegetation is dense, tree ferns rising 8, 10, 12 meters, their fronds casting deep shade over the water's edge. Behind them, conifers of several species, their forms not entirely unlike a modern cypress or araucaria, rising 40 meters or more into the hazy sky. The air smells of resin and wet soil and decaying plant material. It smells, emphatically, of green, of chlorophyll and decomposition, and the particular heavy humidity of a closed forest in warm rain. There is sound, insects, lots of insects, beetles, flies, primitive wasps, dragonflies with wingspans that would give a modern observer pause. There may be small dinosaurs in the undergrowth, feathered, quick, sharp-eyed. Early mammals, nocturnal and cautious, are somewhere in the shadows, probably asleep. The lake itself supports fish, early turtles, and various invertebrates. And there, at the water's edge, half submerged, unremarkable among the tangle of aquatic vegetation, is Archifructus. You would not stop to look at it. It has no petals. It produces no fragrance. Its reproductive structures are small, tightly packed, arrayed along a thin stem in a way that suggests function rather than display. If you were a cretaceous insect and you were searching for food, you might investigate it. You might not. It is not advertising itself with any of the visual or chemical signals that will define its descendants. But inside those small, closed structures, inside the carpels that give this plant its name, ancient fruit, something is happening that has never happened before on Earth. A seed is developing inside an enclosed vessel of the plant's own tissue. Not exposed, not naked, enclosed, protected. And the pollen that fertilized it arrived via a mechanism that will, over millions of years, become the most productive ecological relationship in the history of terrestrial life. Montsechia, the other early angiosperm, lived a similar existence in the freshwater lakes of what is now Spain. Fully aquatic, submerged, without petals, without any of the visual appeal we associate with flowers. It looked, if anything, like a particularly unremarkable aquatic weed. If you had told any observer in that Cretaceous lake that the descendants of this plant would one day cover 80% of all terrestrial plant species, would build the tropical rainforests, would produce every grain of rice and wheat and corn that human civilizations would depend upon, that observer, if they could somehow comprehend the claim, would have found it incomprehensible, possibly offensive in its implausibility. And yet, the implausibility is precisely the point. Evolution does not announce its revolutions in advance. It does not flag its most consequential experiments with drama or scale. The most important biological innovations in the history of life have almost without exception begun in obscurity, small, local, unremarkable, invisible to any observer not equipped with the benefit of 200 million years of hindsight. The first cells, the first multicellular organisms, the first land plants, the first flower. Each one was, at its moment of origin, nothing. A local solution to a local problem. A small increment of reproductive efficiency in a world that was getting along perfectly well without it. What makes the flower different? What makes it worth 90 minutes of your attention is the cascade it eventually triggered. Not immediately, not quickly by any human measure, but with a thoroughness and a reach that no other biological innovation in the history of land life has matched. The geography was about to help it, because the world of 130 million years ago was not stable. It was fragmenting, and fragmentation in evolutionary biology is the engine of diversification. The Tethys Sea, that ancient tropical ocean that stretched in a broad band across the middle of the globe, separating the northern landmasses from the southern, was contracting as continents moved. Its warm, shallow margins created corridors and barriers in shifting combination, isolating populations of plants and animals for millions of years, then reconnecting them, then isolating them again. Each cycle of isolation and reconnection was an opportunity for evolutionary divergence. Each barrier was a filter that selected for organisms capable of crossing it, or capable of surviving on one side of it, or capable of adapting rapidly to conditions on the other. The early angiosperms were, almost certainly, weedy plants, colonizers, opportunists, specialists in disturbed and marginal habitats. The traits that defined them, short generation times relative to conifers, flexible growth forms, efficient water transport, enclosed and protected seeds, were precisely the traits that favor organisms in unstable, frequently disturbed environments. They were the ecological equivalent of the first organisms to colonize a cleared field or a volcanic island. And the mid-Cretaceous world, with its shifting sea levels, its active tectonics, its fragmenting continents and contracting ocean corridors, was producing disturbed and marginal habitats in abundance. The stage was set. The organism was ready. What happened next was not a single event, but a slow, grinding, relentless process of competitive replacement, played out across continents, across millions of years, across thousands of ecological interactions that would end with flowering plants in possession of the terrestrial world. The change begins subtly. In the fossil record, we first detect it not through flowers, nothing so dramatic as a bloom, but through pollen. Tiny grains of pollen recovered from ancient lake sediments in Morocco, in Israel, in what is now Italy and England, dating to approximately 135 million years ago. The grains are distinctive. They have a single furrow, a colpate structure in the technical language of paleontology, that marks them as different from anything that came before. They are angiosperm pollen, the signature of a flowering plant. But the plant itself? Invisible. The body of the plant, if it existed in these localities, left no recoverable trace. We have only the pollen, the biological signal that something new was happening somewhere in the early Cretaceous world. And here is where things get genuinely strange. Here is where the story takes its first unexpected turn. Because when you ask the obvious next question, where did these pollen grains come from? What did the plant look like? What was the ancestor of all flowering plants? The fossil record goes quiet. It offers no answer, or rather, It offers competing, contradictory, maddeningly ambiguous answers. Charles Darwin understood this problem intimately. In 1879, he wrote a letter to his close friend and collaborator Joseph Hooker, the botanist who would become director of Kew Gardens. And in that letter, Darwin used a phrase that has echoed through paleobotany ever since. He called the origin of flowering plants an abominable mystery, not a puzzle, not a question. Abominable. The word choice tells you everything about how much it bothered him. Darwin was a man who had spent decades demonstrating that complex biological structures arise through gradual, incremental change. That there are no sudden appearances, no leaps, no discontinuities that cannot eventually be explained by patient excavation of the fossil record. His entire intellectual edifice rested on gradualism. And flowering plants violated gradualism. They appeared in the Cretaceous fossil record suddenly, abundantly, and in enormous diversity, as if they had been waiting off-stage and simply walked on in full costume, fully formed, already playing a dozen different roles simultaneously. That was 146 years ago, and in the intervening century and a half, we have learned extraordinary things about the origins of flowering plants. We have molecular clocks that can estimate divergence times from genetic data. We have new fossil sites producing remarkable specimens. We have imaging technologies that allow us to see the internal structure of fossils that would have been opaque to Darwin's microscope. We have, in short, vastly more information. And yet, the abominable mystery remains substantially abominable. New research published in 2025 by a team working under the auspices of the Royal Society has confirmed that the disagreement between molecular evidence and fossil evidence on the age of angiosperms is not an artifact of poor methodology. It is a genuine, deep, unresolved tension. Molecular clocks consistently suggest that the ancestors of flowering plants diverged from other seed plants not in the Cretaceous but in the Jurassic, or even earlier. Some analyses push the origin back into the Permian, more than 250 million years ago. But unequivocal fossil evidence for crown angiosperms, plants that definitively share the full suite of characteristics we associate with flowering plants, does not appear before the Cretaceous. Something is missing from the record. The early flowering plants may have existed for tens of millions of years before they appear in the fossil record with confidence. They may have been rare or restricted to upland environments that preserve poorly, or simply unlike what we expect a primitive flowering plant to look like. The ghost lineage, the evolutionary history before fossilization finds it, may be long and complex and, for now, lost to time. This is not a failure of science. This is science working exactly as it should, holding competing hypotheses with intellectual honesty, refusing to collapse ambiguity into false certainty, continuing to search. But it means that when we trace the story of flowering plants, we are tracing a story with a missing first act. We know the revolution happened. We do not yet know with precision where or exactly when it began. What we do know, with confidence, with abundant evidence, with beautiful physical specimens you can hold in your hand, is what the first recognizable flowering plants looked like. And the answer is deeply anticlimactic. and deeply instructive. They were not roses. They were not orchids. They were not the thing your mind conjures when you hear the word flower. The oldest confidently identified angiosperm macrofossils, the actual preserved remains of the plants themselves, not just their pollen, come from two sources, and both are revelatory in their strangeness. The first is Archifructus liaoningensis, recovered from the Yixian Formation in what is now Liaoning Province in northeastern China. The Yixian Formation is a remarkable geological unit, a series of lake sediments laid down approximately 125 million years ago during a period of intense volcanic activity. The same formation has yielded feathered dinosaurs, early birds, and an extraordinary diversity of Cretaceous insects. It is one of the most productive windows into early Cretaceous life on Earth. And from those sediments comes Archifructus. The name means ancient fruit, a deliberate choice that points to what makes this organism significant. Archifructus is aquatic. It grew in shallow water, or possibly along the water's edge. Its reproductive structures, the carpels, the enclosed seeds, are arranged in a way that paleobotanists find consistent with angiosperm identity. But it has no petals. It has no sepals. It has nothing that we would ordinarily call a flower in the aesthetic sense. It is a small, inconspicuous water plant with a specialized reproductive strategy. The second key specimen is Montsechia vidali, recovered from limestone deposits in the Iberian mountains of Spain. Montsechia is also aquatic, possibly a fully submerged plant, living entirely underwater in shallow freshwater lakes. It dates to approximately 130 million years ago, making it one of the oldest known angiosperms. And like Archifructus, it is not beautiful. It is not colorful. It is not, in any conventional sense, what you would point to as the ancestor of a sunflower or an apple tree. And yet, these humble, water-dwelling, petal-less organisms represent the opening movement of a biological symphony that would, over the following 50 million years, come to dominate every terrestrial ecosystem on Earth. Why? Why did these particular plants, with this particular reproductive strategy, eventually win so thoroughly? The answer lies not in the flower as we see it, not in the petal, the color, the fragrance, but in the machinery hidden inside. The structural innovations that define an angiosperm are not superficial. They are deeply, fundamentally mechanical. They represent a reorganization of how reproduction works at the most basic level. Let us start with the carpel. The carpel is the enclosed seed vessel, the structure that contains and protects the ovule before fertilization and becomes the fruit after. In gymnosperms, in conifers, in cycads, in ginkgos, the seed is naked. It sits exposed on a scale of a cone, accessible to the environment, unprotected by any enclosing structure. The gymnosperm trusts to its seed's own defenses, thick seed coats, resins, chemicals, to survive. The angiosperm does something different. It wraps the seed in tissue. It encloses it. It creates a private chamber, a controlled environment, a sealed vault within which development occurs under the plant's regulation. This is the first revolution: enclosure. The word angiosperm means enclosed seed, from the Greek angion, vessel, and sperma, seed. Everything follows from this one structural commitment. Because once you have enclosed the seed, you have also created the possibility of the fruit. The fruit is nothing more than the matured carpel, the vessel, ripened, transformed, made edible or otherwise attractive to animals. And once you have fruit, you have something to offer. You have a currency. You can trade. The second innovation is equally profound and far less obvious. It is called double fertilization, and it is something that exists nowhere in the plant kingdom outside the angiosperms. It is, as far as we know, a uniquely flowering plant invention. In gymnosperms, fertilization is single. One sperm cell meets one egg cell. One event produces one embryo. The surrounding tissue that feeds the developing embryo, the endosperm, is pre-formed, prepared in advance, regardless of whether fertilization actually occurs. The plant commits its resources speculatively. In angiosperms, something more elegant happens. The pollen tube delivers not one sperm cell, but two. The first sperm cell fertilizes the egg, producing the embryo, the new plant. The second sperm cell fertilizes a separate cell within the ovule, producing the endosperm. Both fertilizations happen simultaneously from the same pollen delivery. The consequence is enormous, because now the endosperm, the nutritive tissue that will feed the embryo, is only produced when fertilization actually occurs. The plant does not have to invest in feeding an embryo that might never exist. Resources are committed conditionally, precisely, efficiently. This is not a minor metabolic saving. Over the course of an evolutionary lifetime, the ability to allocate nutritive resources conditionally rather than speculatively is a profound competitive advantage. And there is a third innovation, less celebrated but equally important: the xylem vessel. The vascular tissue of flowering plants, the internal plumbing through which water and nutrients move, contains a structure called a vessel element that is fundamentally different from the tracheid cells that gymnosperms use for the same purpose. Vessel elements are wider, more open, more efficient at moving water under pressure. They allow flowering plants to move water faster, to support larger leaves with more complex venation patterns, to photosynthesize more efficiently, to grow faster in favorable conditions. The leaf veins of angiosperms are worth pausing on. The reticulate venation, the network of interconnected veins that you can see if you hold an oak leaf up to the light, creates a redundant vascular system. If one vein is damaged or blocked, the network reroutes. Water keeps flowing. In gymnosperms, the veins run parallel, unbranching, efficient but fragile. Damage one pathway, and that part of the leaf dies. The angiosperm leaf is a robust, fault-tolerant hydraulic network. The gymnosperm leaf is a simpler, more vulnerable pipeline. Put these three innovations together: the enclosed seed, double fertilization, and efficient vascular architecture, and you have a plant that is, under the right conditions, more efficient, more flexible, more capable of rapid reproduction, and more capable of entering into productive relationships with animals than anything that came before it, under the right conditions. That qualifier matters enormously, because the flowering plants did not immediately conquer the world. The early Cretaceous landscape was still dominated by gymnosperms. The conifers were not going anywhere. The cycads were not going anywhere. For tens of millions of years, the angiosperms remained a minor component of most floras, ecologically marginal, restricted to disturbed or disturbed adjacent habitats, present in the record but not dominant. And then something happened. Multiple things happened. More precisely, a convergence of geological, climatic, and biological events that opened the door, and the flowering plants walked through it. The first was geography. Pangaea had been breaking apart for millions of years, but in the mid-Cretaceous, roughly 100 million years ago, the process was accelerating dramatically. The South Atlantic was opening. India was separating from Africa and beginning its long northward journey toward the collision that would eventually raise the Himalayas. North America and Europe were pulling apart. The world was becoming an archipelago of separating landmasses, each developing its own climate, its own corridors, its own ecological pressures. This matters for flowering plants in a specific way. Fragmentation creates opportunity. When a single large landmass breaks into multiple smaller ones, populations that were once connected become isolated. Isolated populations evolve independently. Different selective pressures in different environments drive divergence. The result is rapid speciation, the creation of new species at a pace that connected, homogeneous environments cannot produce. The flowering plants, with their shorter generation times than most gymnosperms, their higher reproductive rates, their conditional resource allocation, they were exquisitely positioned to exploit this fragmentation. Where a conifer might require decades to reach reproductive maturity and produce its first viable seed, an early angiosperm might accomplish the same in months or years. Faster reproduction means faster evolution, means faster adaptation to new and changing conditions. The second factor was climate. The mid-Cretaceous was, in many regions, experiencing intense seasonal variation. Wet seasons and dry seasons, flooding and drought. Temperature swings that gymnosperms, adapted to more stable Jurassic conditions, handled less well. Flowering plants, with their deciduous potential, the ability to drop leaves and go dormant during unfavorable periods, a capacity linked to their efficient vascular architecture, could tolerate and exploit seasonal climates that put gymnosperms under stress. And the third factor was biological. It was the most important factor. It was the one that created a feedback loop so powerful that it drove tens of millions of years of co-evolution and ultimately reshaped the living world from the ground up. The third factor was the insects. Now, insects and plants had been interacting for a very long time before flowering plants existed. This is a point that recent research, particularly a major review published in 2025, has been at pains to emphasize. Insect pollination did not begin with flowering plants. Scorpion flies with specialized mouthparts were visiting gymnosperms in the Jurassic. Beetles were feeding on gymnosperm pollen and inadvertently transferring it. The mutualistic relationship between plant and insect was already being sketched out in rough preliminary form long before a single angiosperm existed. But the angiosperm flower represented something qualitatively new in this relationship. It represented the evolution of a deliberate interface, a structure specifically shaped to attract, accommodate, and interact with animal visitors. The petal is a landing pad and a signal. The nectary is a reward. The fragrance is an advertisement broadcast through chemical channels. The color targets the visual systems of specific pollinators. The flower, in other words, is not simply a reproductive structure. It is a communication device. It is a contract written in biology. Consider what this contract achieves. A wind-pollinated gymnosperm releases its pollen in clouds and hopes. A bee-visited angiosperm delivers its pollen with precision. The bee is a targeted delivery vehicle. It visits a flower of one species, picks up pollen, and, crucially, is motivated by its need for nectar and pollen to visit another flower of the same species. The flower's shape, color, and scent are filters. They ensure that the bee is visiting the right species, not any flower that happens to be open. The result is a pollination efficiency that wind pollination cannot approach. This precision has a name in evolutionary biology. It is called pollination syndrome, the set of floral characteristics that co-evolve with a specific type of pollinator to create a mutually beneficial, mutually reinforcing relationship. Blue and ultraviolet flowers with nectar guides attract bees. Red tubular flowers attract hummingbirds, whose red vision exceeds that of most insects. White flowers that open at night and emit heavy, sweet fragrances attract moths. Deep tubular flowers with long spurs attract long-tongued flies, or moths with precisely matching proboscis lengths. Darwin himself provided one of the most dramatic demonstrations of this principle. In 1862, examining a Madagascan orchid, Angraecum sesquipedale, he noted that the flower had a nectar spur approximately 30 centimeters long. He predicted, and was widely mocked for predicting, that there must exist a moth with a proboscis 30 centimeters long that pollinated it. 40 years after Darwin's death, the moth was discovered. Xanthopan morgani predicta, the predicted moth. The prediction was so precise, so testable, so elegantly confirmed that it stands as one of the great demonstrations of evolutionary logic in the entire history of science. But here is where the story takes a turn that the textbooks often understate. The relationship between flowering plants and their pollinators was not a simple, linear co-evolution in which plant and insect locked onto each other and developed together in perfect synchrony. The reality was messier, more surprising, and in some respects more impressive. The 2025 research on deep time pollination ecology makes this clear. There is, as the researchers found, a temporal gap between the appearance of the major pollinator lineages and the main radiation of angiosperms. Bees, butterflies, and many flies diversified not simultaneously with flowering plants, but somewhat after them, tracking the revolution rather than triggering it. The initial explosion of angiosperm diversity in the early to mid-Cretaceous appears to have been driven, at least in part, by older insect groups, beetles, flies, certain wasps, that were already present and already practiced in visiting plants for food. This means that the angiosperm revolution was not a single, unified co-evolutionary event. It was a cascade. Flowering plants diversified using existing, already available insect visitors. As they diversified, they created new niches, new food sources, new habitats, new ecological opportunities. Those new opportunities drove the evolution of specialized pollinator lineages: the bees, the butterflies, the long-tongued flies. Those new pollinator lineages then drove further floral diversification, selecting for ever more specific, ever more elaborate, ever more precise floral forms. And so it went, wave after wave, for tens of millions of years. Let us focus for a moment on the bees, because their story is particularly instructive and, in a deep sense, personal. The first bees appear in the Cretaceous fossil record as something that would have been unrecognizable as a bee to any modern observer. They were wasp-like, slim, predatory in ancestry, derived from a crabronid wasp lineage that hunted other insects and provisioned their nests with arthropods as food for larvae. At some point, one lineage made a dietary switch. Instead of catching prey insects to feed their young, they began collecting pollen. Pollen is protein-rich, abundant, and, importantly, available from flowers that the insect was already visiting for nectar. The switch was not dramatic. It was incremental, but its consequences were immense. Because a pollen-collecting insect is a more efficient pollinator than a predatory one, a bee has evolved specialized structures, the corbiculae, the pollen baskets on the hind legs of most social bees, the dense hairs on the body that trap pollen electrostatically, precisely for the purpose of collecting and transporting pollen. And this is where the physics becomes fascinating. Bees build up a static electric charge as they fly. Flowers develop their own weak electric field. When a charged bee approaches a flower with a different electric polarity, pollen is literally attracted off the flower and onto the bee, drawn by electromagnetic force without physical contact. The flower and the bee have co-evolved to exploit the physics of charged particles. This is not metaphor. This was documented in research from the University of Bristol, and it represents one of the most unexpected and elegant physical mechanisms ever discovered in an ecological relationship. From their wasp-like origins in the early Cretaceous, bees diversified explosively through the late Cretaceous, and especially the Paleogene, tracking and feeding back into the angiosperm radiation in exactly the stepwise, cascading pattern we described. Today, there are approximately 20,000 known species of bees. Almost every one of them depends, directly or indirectly, on flowering plants for survival. And approximately 90% of all flowering plant species require animal pollination to reproduce effectively. The interdependence is so complete, so total, so mutually constitutive that it is difficult to say, at the level of the ecosystem, where the plant ends and the bee begins. They are, in the deepest biological sense, aspects of a single system. The relationship between flowering plants and their pollinators is, in the popular imagination, a story of beauty and harmony. The bee visits the flower rewards the bee. Both flourish. It is a story that lends itself to metaphor, to poetry, to the kind of warm ecological narrative that makes nature documentaries feel reassuring. The reality is considerably stranger and considerably more interesting. Because the flower is not simply a reward system, it is, in many cases, a manipulation system, a deception machine, a biological con artist of extraordinary sophistication that has, in certain lineages, abandoned the pretense of offering any reward at all, and instead evolved to exploit the perceptual systems of its pollinators through pure trickery. The orchids are the masterclass in this. With approximately 28,000 known species, the Orchidaceae is the largest family of flowering plants on Earth, and a disproportionate number of those species are, in one way or another, cheating. Approximately 1/3 of all orchid species offer their pollinators no reward whatsoever, no nectar, no pollen worth collecting, nothing. They have evolved flowers that resemble, with varying degrees of accuracy, something that their target pollinators want: food, a mate, a safe resting place, without providing it. The pollinator is lured in, picks up or deposits pollen while attempting to obtain something that does not exist, and leaves, tricked, manipulated, used. The most extraordinary version of this deception is sexual mimicry, and the bee orchids of the genus Ophrys are its most celebrated practitioners. These plants, distributed across the Mediterranean and into Central Europe, have evolved flowers that mimic, in extraordinary detail, the appearance, the texture, and the scent of female bees and wasps. The lip of the flower, the labellum, has the coloration, the surface texture, even the fine velvety hairs of a female bee. But it is the chemistry that is truly staggering. The orchid synthesizes a blend of chemical compounds, hydrocarbons, aldehydes, alkenes, that precisely mimics the sex pheromones of a specific species of female bee or wasp. The male bee, responding to what his olfactory system tells him is an unmated female, attempts to mate with the flower. During this pseudo-copulation, a term that is exactly as undignified as it sounds, the male contacts the sticky pollen masses of the orchid, called pollinia, which attach to his body. He eventually realizes his mistake, disengages, and flies away. He will, with some probability, make the same mistake again at another flower of the same species, and in doing so, transfer the pollen. The orchid has evolved to exploit a fundamental feature of animal neurology: the fact that sensory systems are not perfect. They are tuned to detect the most important signals in the environment, and they can be fooled by a sufficiently accurate imitation. The bee's brain did not evolve to distinguish orchid from female bee. It evolved to detect female bees efficiently. The orchid has reverse engineered that detection system and turned it into a delivery mechanism for pollen. What makes this even more remarkable is the specificity. Each Ophrys species typically mimics one specific bee or wasp species. The chemical blend is tuned to that species' pheromone profile. The flower color and shape match that species' visual profile. The result is a relationship of extreme specificity. One orchid, one pollinator, zero reward. That has evolved independently many times across the orchid family and persists because, despite the lack of reward, the system works. The male bee is fooled often enough that pollen transfer occurs, seeds are set, and the orchid reproduces. Darwin spent years studying orchids. He found them, in a letter to a colleague, more interesting than any other group of organisms he had examined in the context of natural selection, precisely because they demonstrated so vividly that natural selection is not a process aimed at beauty or harmony or mutual benefit. It is a process aimed at reproductive success. Whatever achieves that goal persists, regardless of whether it involves beauty, deception, exploitation, or anything in between. But deception is only one end of the spectrum. At the other end is something equally extraordinary: the chemistry of the fruit. We have spoken of fruit as reward, as currency, as the angiosperms' gift to its seed dispersers. And that framing is accurate as far as it goes. But it is incomplete, because the fruit is not simply a passive offering. It is a precisely engineered biological product that controls, with considerable sophistication, who eats it, when they eat it, and what happens after they eat it. Consider the color change of ripening fruit. A fruit begins green, the color of leaves, invisible against foliage, indistinguishables to most animals and therefore protected against being eaten too early, before the seed is viable. As the seed matures, the fruit changes color: red, orange, yellow, blue, black. The specific color it changes to is, in many cases, tuned to the visual system of the intended disperser. Fruits dispersed primarily by birds tend to turn red or bright orange, colors that bird tetrachromatic vision detects with exceptional sensitivity, but that are poorly visible to mammals with dichromatic or trichromatic color vision. Fruits dispersed by mammals, including primates, tend to turn yellow or orange, colors well within the range of primate trichromatic vision. The color change is a signal. I am ready. Come now. The seed will survive your digestive system. There is a reward waiting. But the fruit also contains, very often, a chemical arsenal directed at the wrong visitors. Animals that would destroy the seed rather than disperse it. Tannins, which make unripe fruit astringent and unpleasant to eat. Alkaloids, caffeine, nicotine, capsaicin, morphine, many of which are, in high doses, toxic to insects and small mammals, but are either tolerated or actively sought by larger animals. The Chile pepper produces capsaicin that burns mammalian mouths. The capsaicin receptor in mammals responds to it as a pain signal. But birds, which are the intended seed dispersers of many chili species, lack this receptor entirely. The pepper is simultaneously repellent to the wrong disperser and attractive to the right one. The chemistry is precise. The targeting is specific. Caffeine is produced by coffee plants, tea plants, and several other angiosperms. And its primary ecological function is almost certainly not to produce a pleasant morning beverage for primates. It is a neurotoxin, a compound that disrupts the adenosine receptors of insects that feed on the plant's leaves and seeds. At low doses, interestingly, caffeine has been shown in recent research to improve the memory of bees visiting caffeinated flowers. They remember the flower's location better and return more reliably. The plant has accidentally, or perhaps not so accidentally, discovered a way to improve the performance of its pollinator by dosing it with a neurologically active compound. The flower as coffee shop, rewarding its regulars with a mild cognitive enhancer. The chemical complexity of the angiosperm is staggering. Flowering plants collectively produce an estimated 200,000 distinct secondary metabolites, chemical compounds that are not directly involved in basic metabolism but serve ecological functions. Defense against herbivores, attraction of pollinators, deterrence of the wrong seed dispersers, signaling to mycorrhizal fungi in the soil, communication through volatile compounds released into the air with neighboring plants about herbivore attack. The angiosperm is not a passive organism waiting to be eaten or pollinated. It is a chemical factory of extraordinary output, running a continuous multi-channel chemical conversation with every organism in its vicinity. This chemical complexity is, among other things, the basis of human pharmacology. Approximately 50% of all pharmaceutical drugs either are derived from plant compounds or were developed using plant compounds as molecular templates. Aspirin from willow bark, morphine from the opium poppy, quinine from cinchona bark, Taxol, one of the most effective cancer chemotherapy agents known, from the Pacific yew tree. Artemisinin, the most effective treatment for malaria, from sweet wormwood. The chemical defenses that angiosperms evolved over 100 million years of coevolutionary arms races with insects and herbivores are the same compounds that human biochemists, working backwards from the observation that certain plants have physiological effects, have turned into the pharmaceutical arsenal of modern medicine. The flower's chemistry is not decoration. It is not coincidence. It is the product of 100 million years of molecular evolution, of selection pressure applied generation after generation to every compound a plant produces, in every ecological context the plant inhabits. The result is a library of biological active molecules of a breadth and sophistication that no human chemist working from first principles could have designed. We are still in 2025. Discovering new compounds in flowering plants that have medical significance. The library is not fully catalogued. It may never be. By approximately 100 million years ago, the mid-Cretaceous, the Cenomanian age, something in the fossil record changes decisively. The pollen curves shift. In sediment cores from this period, the proportion of angiosperm pollen relative to gymnosperm pollen begins to rise, slowly at first, and then more sharply. The leaf floras of late Cenomanian and Turonian deposits show angiosperms not as rare curiosities, but as common, dominant elements. And critically, the diversity of angiosperm forms begins to explode. Magnolias, laurels, sycamores, roses, or at least their early precursors, water lilies, monocots, the group that will eventually produce grasses, palms, and orchids. The body plan of the flowering plant is diversifying across every ecological niche available to it. Low herbs colonizing disturbed soils, climbing vines exploiting the vertical architecture of gymnosperm forests, shrubs occupying the forest edge, and the first woody angiosperms, the ancestors of the great broad-leaved trees that will eventually dominate temperate and tropical forests worldwide. The woody angiosperms are worth special attention because they represent a qualitative change in what the angiosperm revolution could accomplish. An herbaceous annual plant, a small flowering herb that lives for one season, can evolve quickly, reproduce abundantly, and colonize open habitats with impressive efficiency. But it does not build forests. A woody angiosperm does. It creates structural complexity. It builds canopies. It produces the architectural scaffolding within which an entire community of organisms, insects, birds, mammals, fungi, other plants, can find shelter, food, and ecological purpose. The transition from gymnosperm-dominated forests to angiosperm-dominated forests was not a sudden replacement. The conifers did not die. They retreated to high altitudes, to high latitudes, to nutrient-poor soils and exposed ridgelines where their particular physiological tolerances gave them a competitive edge that broad-leaved angiosperms could not overcome. They survive in those refugia to this day. The boreal forests of Canada and Russia, the taiga, stretching across the northern continents in an almost unbroken belt, are gymnosperm forests, vast and ancient and entirely functional. But south of the taiga, in the temperate zones and especially in the tropics, the angiosperm revolution was total. And here we arrive at what scientists have now officially termed the angiosperm terrestrial revolution, the ATR. This is the name that a group of researchers led by Michael Benton of the University of Bristol proposed in 2022 in a landmark paper in the journal New Phytologist. And it is the framework that all subsequent work on this topic has adopted. The ATR spans, in Benton's formulation, from approximately 100 million years ago to approximately 50 million years ago, from the first high-diversity angiosperm floras in the mid-Cretaceous to the establishment of fully modern, angiosperm-dominated tropical forests in the early Paleogene. 50 million years. 100 million years of relentless biological transformation. And the ATR did not merely change the plants. That is the finding that makes this story truly staggering in its scope. The ATR reset the entire terrestrial biosphere. It was not a plant revolution that happened while the animals watched. It was a revolution in the fundamental productivity, structural complexity, and ecological architecture of land, which then drove the diversification of essentially every major group of land animals that matters to the modern world. Let us count them. Let us go through the roll call of organisms whose evolutionary history was either created or dramatically accelerated by the rise of flowering plants. Insects first, since we have already begun that story. The ATR drove the diversification of beetles, already the most species-rich order of animals before the revolution, but made dramatically more so by the new food sources, new habitats, and new ecological niches created by flowering plants. It drove the radiation of the Lepidoptera, the butterflies and moths, whose caterpillars feed primarily on angiosperm leaves, and whose adults pollinate angiosperm flowers. It drove the diversification of wasps, ants, and the rest of the Hymenoptera, many of which are intimately associated with flowering plants in pollination or in herbivory, or in the complex parasitic relationships that build up around any abundant food resource. It drove the diversification of flies, the Diptera, many of whose larvae feed on the fallen organic matter of angiosperm forests, and whose adults are significant pollinators. The result is that insects, already the most diverse animals on Earth in terms of species number, became more diverse still, in direct proportion to the diversification of flowering plants. The two curves track each other through the late Cretaceous and Paleogene with a fidelity that is not coincidence. It is causation. The plant creates the niche. The insect fills the niche. The insect drives further plant diversification by becoming a more specialized pollinator. The plant creates new niches for new specialists, and so on, in a diversification spiral that lasted 50 million years, and whose products you can observe in your garden on any warm summer afternoon. Spiders next. This may seem surprising. Spiders are predators, not pollinators, not herbivores. What do they have to do with flowering plants? The connection is indirect but powerful. Spiders eat insects. As insects diversified in the ATR, becoming more abundant and more diverse, the ecological opportunity for insect predators expanded correspondingly. Spiders radiated extensively through the Cretaceous and Paleogene, and the web-spinning lineages, the araneomorphs, which build the flat orb webs and three-dimensional cobwebs that are the most familiar to us, diversified in particular. Their prey was, in large part, the insects of the angiosperm world. Birds. The story of birds and the ATR is one of the most striking in the whole narrative. Birds evolved from theropod dinosaurs in the Jurassic. Archaeopteryx, the famous transitional fossil, dates to approximately 150 million years ago. Through the Cretaceous, while dinosaurs still walked the earth, birds were diversifying slowly, experimentally, filling niches in trees and cliffs and water margins. But the great radiation of modern bird groups, the explosion of species diversity that produced the tens of thousands of bird species alive today, happens in the Paleogene after the extinction of non-avian dinosaurs, and it tracks the angiosperm revolution with remarkable fidelity. Why? Because flowering plants produce fruit, and fruit is, for birds, the most transformative food source imaginable. Fruit is energy-dense, abundant, and produced by plants in a structure deliberately evolved to be eaten and the seeds to be carried away. The plant and the bird are in a contract. The plant invests calories in producing a nutritious, visually attractive fruit. The bird eats the fruit and deposits the intact seed, packaged in its own fertilizer, at some distance from the parent plant. Both parties benefit. The plant gets seed dispersal. The bird gets calories. This fruit-bird contract drove the evolution of fruit-eating specialists, birds with wide gapes for swallowing large fruits whole, birds with gut chemistry that can handle the secondary compounds in certain fruits, birds with color vision tuned to detect ripe fruits against a background of green leaves. Red fruits, blue-black fruits, orange fruits, all are colored to be maximally detectable by bird color vision, which extends into the ultraviolet in ways that mammal vision cannot match. The flower advertises to its pollinator. The fruit advertises to its disperser. In both cases, the advertisement is a signal co-evolved with the perceptual system of the intended recipient, and mammals. Here the story becomes, for us, most personal. Our own lineage, the mammals, existed before the angiosperm revolution. The earliest mammals appear in the Triassic, alongside the first dinosaurs. Through the entire Jurassic and most of the Cretaceous, mammals remained small, primarily nocturnal or crepuscular, ecologically marginal. The dinosaurs occupied the large body, daytime ecological natures. Mammals occupied the gaps. But through the Cretaceous, as angiosperms spread and diversified, something was quietly changing for the mammals. Fruits appeared, seeds appeared, nuts appeared. High energy, protein-rich, seasonally abundant food sources that small, warm-blooded, metabolically expensive animals, which is what early mammals were, could exploit more efficiently than large, cold or cool-blooded dinosaurs. The seeds and fruits of the early angiosperms were, for early mammals, a nutritional windfall, a new food category that had not previously existed. The angiosperm fossil record shows a clear increase in the number of plant species producing fleshy fruits and seeds from the late Cretaceous onward. The mammal fossil record shows, in parallel, a clear increase in dental complexity. The molariform teeth that allow the efficient processing of hard, resistant food items like seeds, or the cusped molars adapted for crushing and shearing fruit. The mammalian jaw and the angiosperm fruit are co-evolutionary products. They shaped each other. There is a second wave to the angiosperm revolution, quieter in its beginnings than the first, less dramatic in its visual signature, but in terms of its ultimate ecological impact, arguably equal to everything that came before it. It is the story of grass. Grass is an angiosperm. This seems, stated plainly, almost too obvious to mention. Of course, grass is a flowering plant. But the implications of that fact played out across the Eocene, Oligocene, and Miocene epochs, reshaped the terrestrial world in ways that rival the original Cretaceous radiation. The first grasses appear in the fossil record, primarily as pollen and phytoliths, the tiny silica bodies that grass cells deposit within their tissues and that preserve extraordinarily well in sediment. In the late Cretaceous, perhaps 65 to 70 million years ago, they are present but minor. In the Paleocene and Eocene, they are still marginal elements of forest understories and disturbed habitats. The world of the early Paleogene is a world of forests. Warm, wet, angiosperm-dominated forests stretching from pole to pole, with the early Eocene climatic optimum producing the warmest global temperatures of the past 66 million years. And then the climate cools. Beginning in the Middle Eocene, approximately 40 million years ago, global temperatures begin a long, stepwise decline. Antarctica, now fully separated from South America by the opening of the Drake Passage, begins to accumulate permanent ice. The ocean circulation patterns change. The world becomes, gradually and then more decisively, drier and more seasonal. Forests need rain, consistent year-round rain, or at least a wet season long enough to maintain a closed canopy. As the climate becomes drier and more seasonal through the Oligocene and into the Miocene, the forests retreat. They retreat toward the equator, toward the mountain ranges where moisture is captured by orographic rainfall, toward the coastlines where oceanic humidity maintains precipitation, and into the spaces the forests vacate, into the vast continental interiors of Africa, Asia, North and South America. The grasses move. The grass plant is a masterpiece of engineering for open, dry, seasonally stressed environments. It grows from the base, not the tip, a growth pattern called intercalary growth. That means a grazed grass plant regrows immediately from undamaged tissue at the stem's base. A conifer that is browsed loses its growing tip and dies or is severely stunted. A grass that is grazed simply regrows. This single architectural feature makes grass essentially unkillable by herbivory in ways that no previous plant lineage had achieved. It is an evolutionary solution to the oldest problem in plant-animal interaction. How do you survive being eaten? The grass also produces, in its leaf tissue, microscopic particles of silica, phytoliths, that are, to the teeth of any animal attempting to eat the grass in quantity, essentially sandpaper. Grinding silica-rich grass wears down teeth with extraordinary efficiency. Any large herbivore that wants to subsist on grass needs teeth that can handle this. And so, through the Miocene, we see in the fossil record one of the most dramatic evolutionary responses in vertebrate history: the development of hypsodonty, high-crowned teeth, in a wide range of large mammalian herbivores across multiple continents simultaneously. Horses are the textbook example. The early Eocene horse, Hyracotherium, sometimes called the dawn horse, is a small forest browser, roughly the size of a fox, with low-crowned teeth suited for eating soft leaves and fruits in a forested environment. Through the Eocene and Oligocene, as forests persist, horse evolution is relatively modest. But in the Miocene, as grasslands expand, the horse lineage transforms almost explosively. Body size increases, the number of toes reduces, from 4 on the front feet to eventually 1. The single hoof of the modern horse, the perfect running machine for open plains. And the teeth change dramatically. The molars become high-crowned, elaborately ridged, capable of grinding silica-rich grass for a lifetime without wearing down to uselessness. The grassland horse is not a modified forest horse. It is a new ecological design, produced by tens of millions of years of selection pressure applied by the grass itself. The grass shaped the horse. The grass shaped the bison, the wildebeest, the zebra, the antelope, the gazelle, the pronghorn, every great grazing animal of every continent's open plains, by demanding, through the physics of silica grinding and the biology of regrowth, a specific set of anatomical solutions that convergently appeared independently in multiple mammalian lineages on multiple continents. And the grazers shaped the grass in return. Grazing prevents the establishment of tree seedlings. A grass-covered plain is maintained as a plain partly by the constant browsing pressure of large herbivores that prevent any woody plant from growing tall enough to shade out the grass. The herds and the grassland are a self-reinforcing system, each maintaining the conditions the other requires. The grasslands also change the atmosphere. Grasses photosynthesize via a biochemical pathway called C4 photosynthesis that is more efficient than the C3 pathway used by most other plants, including most trees, under conditions of high temperature, high light, and low atmospheric carbon dioxide. As atmospheric CO2 declined through the Miocene, the C4 grasses gained a competitive advantage over C3 vegetation that accelerated their spread. The grasslands expanded in part because the global carbon cycle was creating chemical conditions that favored them over forests. And as they expanded, they altered the carbon cycle further. Grassland soils store carbon differently than forest soils, and the global spread of grasslands had measurable effects on atmospheric composition. We are, right now, living inside the ecological legacy of the grass revolution. The great cereal crops, wheat, rice, corn, barley, oats, sorghum, millet, are all grasses. Every loaf of bread, every bowl of rice, every tortilla, every pint of beer is a product of grasses that are themselves the product of the Miocene climatic shift and the angiosperm revolution that preceded it. The human population of 8 billion people is fed primarily by 6 grass species. The entire edifice of agricultural civilization rests on the photosynthetic output of the grass family, the Poaceae, which exists because the climate cooled, the forests retreated, and one lineage of flowering plants was exquisitely positioned to fill the space that opened. The mammoth steppe, the vast grassland ecosystem that covered much of the northern hemisphere during the Pleistocene ice ages, supporting woolly mammoths, woolly rhinoceroses, cave lions, giant ground sloths, and the horses and bison that would eventually be domesticated by early humans, was a grass ecosystem. The African savanna, with its extraordinary megafauna, is a grass ecosystem. The American Great Plains, which supported an estimated 30 to 60 million bison before European colonization, is a grass ecosystem. The Eurasian steppes, across which horse-mounted civilizations rose and fell across 3,000 years of history, are grass ecosystems. Grass did not merely feed the world. Grass structured the world in which human prehistory unfolded. The open landscapes that our ancestors walked out of Africa across, following herds of grazing animals, were grass. The first domesticated crops were grasses. The first fermented beverages, barley beer, rice wine, were grasses. The first animal domesticates, cattle, sheep, goats, horses, were either grazers of grass or grain-fed on grass seeds. The entire transition from hunter-gatherer to agricultural civilization was, at its material foundation, a transition from foraging in angiosperm forests and grasslands to deliberately cultivating the most productive angiosperm species and concentrating their output. The second wave of the angiosperm revolution did not announce itself with dramatic fossil specimens or the discovery of the world's first flower. It announced itself in pollen samples and phytoliths and the gradual reshaping of continental interiors over tens of millions of years. But its consequences were, if anything, more directly relevant to the emergence of human civilization than the original Cretaceous radiation. Without the first flower, there is no tropical forest, no fruit, no diversification of the mammals that eventually produced the primates. But without the grass, there is no open landscape, no grazing megafauna, no cereal crop, no city, no civilization as we know it. The angiosperm revolution had two acts. We are living inside both of them simultaneously. And then, 66 million years ago, a rock 10 to 15 kilometers in diameter, traveling at approximately 20 kilometers per second, struck the shallow carbonate platform of what is now the Yucatan Peninsula of Mexico, the Chicxulub impactor. The impact released energy approximately 10 billion times greater than the nuclear weapon dropped on Hiroshima. It ignited global wildfires. It injected billions of tons of sulfur aerosols and dust into the stratosphere. It produced an impact winter that lasted years, perhaps decades. A darkness and cold that crashed photosynthesis, collapsed food webs, and drove the most dramatic biotic crisis since the end. Permian extinction 252 million years earlier. The non-avian dinosaurs did not survive. Neither did the marine reptiles, the mosasaurs and plesiosaurs. Neither did the ammonites. Neither did many lineages of birds, insects, and mammals that had evolved in the Cretaceous world. The end Cretaceous mass extinction, the KPG event, named for the boundary between the Cretaceous and Paleogene, eliminated approximately 3/4 of all species on Earth. What happened to the plants? This is a question of intense current research, and the answers are both devastating and, in a deep sense, hopeful. Recent work, incorporated into the 2025 review of the ATR, has confirmed that the impact was catastrophic for Cretaceous plant communities. In well-dated fossil floras from the Americas, more than 50% of plant species went extinct at or near the KPG boundary. Similar patterns have been identified in New Zealand, Europe, and Antarctica. The world that angiosperms had spent 60 million years building was, in significant part, destroyed in years. The answer lies in something the angiosperms had been quietly accumulating for 60 million years. A capability that had nothing to do with flowers or pollinators, and everything to do with what happens when the forest disappears. And when the light returned, gradually, fitfully, over years and decades, it was the angiosperms that responded fastest. Their shorter generation times, their flexible growth forms, their ability to colonize disturbed open habitats, all the attributes that had made them pioneers in the Cretaceous, now made them the first rebuilders of the post-impact world. The weedy, fast-growing, sun-loving angiosperm pioneers flooded the devastated landscapes. The gymnosperms, slower growing, later maturing, less capable of colonizing disturbed soil, recovered more slowly or not at all in many regions. The KPG extinction did not end the angiosperm revolution. It accelerated it. Because the extinction of non-avian dinosaurs did something else. It removed the large terrestrial herbivores that had, for 100 million years, been the dominant consumers of plant material on land. The sauropods were gone. The hadrosaurs were gone. The horned ceratopsians were gone. The browsing pressure that these enormous animals had exerted on the vegetation, and the evidence suggests it was substantial, was removed almost overnight in geological terms. The forests could grow differently. The ecological landscape, suddenly emptied of its dominant consumers, was open to occupation by whatever could exploit it. What could exploit it were the mammals. The Paleogene mammalian radiation, one of the most dramatic evolutionary events in the history of vertebrates, took place in an angiosperm world, feeding on angiosperm food, sheltering in angiosperm forests, adapting to angiosperm-structured ecological niches. Horses, whose ancestors were small forest browsers in the early Eocene, eventually evolved to graze the grass, itself an angiosperm, of open plains. Whales, whose ancestors were terrestrial mammals in the early Eocene, transitioned back to the sea, in part because the productivity of coastal marine systems was recovering and offering new opportunities. Bats, which first appeared definitively in the Eocene, evolved in a world of angiosperm flowers and fruits and became major pollinators and seed dispersers, extending the angiosperm revolution into the night. Primates, our own order. The first primates appear in the fossil record at the very beginning of the Paleogene, immediately following the KPG extinction, in the same Paleocene landscapes that were being rebuilt by angiosperm pioneers. The earliest primates were small, arboreal, probably insectivorous, but they were living in angiosperm trees. And through the Paleocene and Eocene, as those trees diversified and produced new fruits, new flowers, new seeds, the primates that fed on those resources diversified in turn. The primate visual system, our forward-facing eyes, our color vision, our ability to judge distance with precision, is widely thought to have evolved, at least in part, in the context of navigating the three-dimensional complexity of angiosperm forest canopy and detecting the red and orange colors of ripe fruit against green foliage. Our hands, with their grasping capability and fine motor control, are hands shaped by millions of years of reaching for fruits and flowers in the branches of angiosperm trees. The cerebral expansion that eventually produced the human brain has deep roots in the cognitive demands of a frugivorous, arboreal existence in a complex, angiosperm-structured environment. We are, in the most literal biological sense, products of the flower. And here the story reaches a scale that I think deserves a moment of quiet. Not dramatic pause for effect, but genuine contemplative stillness. Because we are talking about something that began as a handful of pollen grains in a Moroccan rock formation 135 million years ago. Something that looked, in its first recognizable form, like a small, unremarkable water plant with no petals, and that eventually produced, through a cascade of ecological interactions and evolutionary feedback spanning 50 million years, the tropical rainforests, the temperate broad-leaved forests, the grasslands, and most of the animals that inhabit them, including the animals watching this video. The numbers, when you state them plainly, are almost incomprehensible. There are approximately 350,000 known species of flowering plants, by far the most species-rich group of land plants on Earth, accounting for approximately 80% of all plant species. Every major terrestrial biome outside the boreal and alpine zones is angiosperm-dominated. Every agricultural staple food, rice, wheat, corn, soybeans, potatoes, sugarcane, is an angiosperm. Every broad-leaved tree in the forest outside your window is an angiosperm. Every grass, every herb, every vegetable, every fruit. Approximately 90% of all flowering plant species depend on animals for pollination, primarily insects, primarily bees. The estimated economic value of pollination services to global agriculture is measured in hundreds of billions of dollars annually. Not because economists have decided to put a number on nature, but because when you remove the pollinators, as we are, in various ways, currently doing, the agricultural system that feeds 8 billion human beings begins to fail in ways that are not easily reversible. The revolution is not history. It is the present tense. But there is a deeper philosophical point here, one that I want to make carefully because I think it matters. The rise of flowering plants is often told as a story of success, of a superior biological innovation outcompeting less efficient predecessors and taking over the world. And in one sense, that is accurate. The angiosperms are, by the usual metrics of evolutionary success, species diversity, geographic range, ecological dominance, the most successful group of land plants in the history of life. But the story of the flower is not really a story about individual organisms competing with each other. It is a story about relationship, about the emergence of a new kind of ecological architecture, one based not on independent self-sufficiency, but on elaborate, mutually reinforcing interdependence. The flower exists because the insect exists. The fruit exists because the bird exists. The forest exists because the mycorrhizal fungi in the soil exist. The underground networks of fungal threads that connect angiosperm roots, transferring nutrients and chemical signals between trees in ways that we are only now beginning to understand. The bee needs the flower. The flower needs the bee. That this appears obvious, inevitable, natural, is itself evidence of how completely the revolution restructured the world around its logic. Darwin called their origin abominable. I think what he meant, beyond the frustration of the missing fossil record, was something closer to awe. The origin of flowers is abominable, in the sense that certain sublime things are abominable, too large, too deep, too consequential to process without a certain vertigo, standing at the edge of something that changes everything, and not quite being able to see where it started. The world that existed in the immediate aftermath of the Chicxulub impact is almost impossible to hold in the imagination without a certain visceral discomfort. Not because of the violence of the impact itself, that was over in minutes, in geological terms, but because of what came after. The years, the decades, the long, cold, dark aftermath. The sulfur aerosols injected into the stratosphere by the vaporization of the carbonate and evaporite rocks of the Yucatan platform. An estimated 100 billion tons of sulfur dioxide, combined with the dust and soot from global wildfires, to produce a stratospheric veil that reduced incoming solar radiation by perhaps 70 to 80% for months, and by lesser but still significant amounts for years. Photosynthesis collapsed, not universally and not instantly, but systematically and globally. The primary producers, the plants and algae that form the base of every food web on Earth, lost their energy source. The food webs they supported began to unravel from the bottom. In the forests of the late Cretaceous, the canopy trees, which had, by 66 million years ago, already been largely replaced by angiosperms across the lower and middle latitudes, began to die. Not from fire, though fires were widespread in the immediate aftermath. From darkness and cold. Photosynthesis requires light. Without adequate light, even a healthy tree cannot maintain itself. Leaves that died were not replaced. Stored energy reserves, the starches and sugars accumulated over decades of growth, were drawn down and exhausted. The great angiosperm forests of the late Cretaceous began, across seasons and years, to fade. The fossil pollen record from KPG boundary sediments in the Americas tells this story with terrible clarity. In section after section, from New Mexico to Colombia to Patagonia, the same pattern appears. Below the boundary, abundant, diverse angiosperm pollen, representing the full complexity of a late Cretaceous forest. At the boundary, a layer, sometimes centimeters thick, sometimes less, sometimes marked by the iridium anomaly that was the first physical evidence of the impact, in which pollen disappears and is replaced by fern spores. Ferns, vast quantities of fern spores, representing what paleobotanists call the fern spike. A world emptied of its forests and temporarily colonized by opportunistic ferns, the fastest colonizing vascular plants, the first responders of the post-apocalyptic landscape. And then, above the fern spike, the angiosperms return, slowly, haltingly, first as weedy, small-leafed pioneers, then as shrubs, then, over hundreds of thousands of years, as forests. Not the same forests. Different forests, with different species compositions, having passed through a bottleneck that eliminated more than half of all plant species in some regions. But angiosperm forests. The revolution, interrupted, resumes. How? The answer is the seed bank, and it is one of the most remarkable adaptive features in the history of terrestrial life. Seeds are dormant. This seems trivially obvious, but its implications are profound. A seed is a plant in suspended animation, metabolically minimal, encased in a protective coat, carrying within it both the genetic information for a new organism and a small but critical endowment of stored energy. Many angiosperm seeds can remain viable in the soil for months, years, or even decades, while conditions are unfavorable for germination. They wait. They do not starve because their metabolic rate is so low as to be nearly unmeasurable. They do not freeze because their seed coats provide thermal insulation and their low water content resists ice crystal formation. They do not rot easily because their coats are often chemically resistant. The gymnosperm seeds on the forest floor and in the cones of standing dead trees also survived in some quantity. But the angiosperm seed bank, the accumulation of seeds deposited in the soil over decades and centuries of forest productivity, was by 66 million years ago a vast distributed repository of genetic information and biological potential. It was, in effect, a biological archive. When the canopy died, when the forest disappeared, when the light came back, fitfully, over months and years as the stratospheric veil thinned and dispersed, the archive opened. The first light of the post-impact recovery fell on a landscape that appeared, from a distance, dead, bare soil, standing dead trees, bleached and bare, ash. The debris of a world that had been burning and dying for years. But in that soil, invisible, patient, metabolically quiescent, were millions upon millions of angiosperm seeds. And some of them, as the light and warmth returned to threshold levels, germinated. The first post-impact angiosperms were not the great trees of the late Cretaceous forests. They were weeds, pioneer species, small, fast-growing, sun-loving, disturbance-adapted plants that could germinate in bare soil, grow to reproductive maturity quickly, and set seed before conditions deteriorated again. Exactly the ecological type that the angiosperms had always excelled at producing. The traits that had made them colonizers and opportunists in the Cretaceous, short generation times, flexible growth forms, efficient reproduction, were precisely the traits that made them the first rebuilders of the post-impact world. The mammals that survived the extinction were, by and large, the same ecological profile: small, adaptable, capable of exploiting whatever food sources were available in a drastically impoverished landscape. Insects, fungi, which bloomed explosively in the post-impact landscape, feeding on the vast quantities of dead organic matter. Seeds from the seed bank, which were accessible to any small mammal with the right dentition. The survivors were generalists and the angiosperms that recovered around them were the generalists of the plant world. Two communities of survivors rebuilding the world together on the debris of the one that had been destroyed. The recovery was not fast by human standards. The full restoration of diverse, complex, angiosperm-dominated forests took millions of years. But in the Paleocene and Eocene, the pace of biological recovery was, by the standards of post-extinction recoveries in the geological record, remarkably rapid. By the early Eocene, approximately 55 million years ago, tropical forests of a complexity and diversity approaching those of today had established themselves across the equatorial regions. By the Eocene climatic optimum, 50 to 52 million years ago, the warmest period since the impact, angiosperm forests stretched from the tropics into the high latitudes of both hemispheres. This is the moment that Benton and colleagues identify as the culmination of the angiosperm terrestrial revolution. Not the first flower, not the Cretaceous radiation, not the survival of the KPG extinction, but the Eocene, the moment when angiosperm-dominated, hyperdiverse tropical forests achieved global dominance, when the mammalian radiation had produced most of the major orders that exist today, when birds had diversified into thousands of species, when insects had achieved something close to their modern diversity. The world, by the early Eocene, looked, in its broad ecological architecture, recognizably like the world we inhabit. It had taken approximately 85 million years from the first Cretaceous angiosperm pollen. 85 million years of continuous, cascading, mutually reinforcing biological transformation. And the result was the biosphere, the living skin of this planet as it currently exists, with all its complexity and productivity and interdependence. We are the latest chapter of that story, not the final chapter. Evolution does not have final chapters, but the latest. The primates that emerged in the Paleocene forests, feeding on angiosperm fruits and seeds, navigating the three-dimensional complexity of angiosperm forest canopy, developed over 60 million years the visual acuity, the manual dexterity, the social complexity, and the cognitive capacity that eventually produced, in one lineage in Africa, the genus Homo. We walked out of angiosperm forests onto angiosperm grasslands. We followed herds of animals that grazed on angiosperm grasses. We learned to cultivate angiosperm seeds. We built our first cities in the river valleys of the Middle East and South Asia and China, in landscapes shaped by angiosperm vegetation, fed by angiosperm crops, organized around the management of angiosperm agricultural productivity. Every civilization in human history has been an angiosperm civilization. Not metaphorically, literally. The material foundation of every human society that has ever existed. The food, the medicine, the timber, the fiber, the spice, the dye, the perfume, the fermented beverage, the domesticated animal fed on grass and grain, is angiosperm in origin. The cotton in your shirt, the wood in your floor, the coffee in your cup, the bread on your table, the aspirin in your medicine cabinet, the oak tree outside your window, the lawn beneath it, all of it. Every material thing that the natural world provides to human civilization, with the exception of fish, of meat from marine mammals, of mineral resources, and of a handful of non-angiosperm timber species. All of it is the product of the revolution that began with an unbeautiful water plant in a Cretaceous lake 130 million years ago. That is not a poetic exaggeration. It is a precise accounting of biological history. We are still, in 2025, arguing about exactly when the first angiosperm evolved. The molecular clock and the fossil records still disagree. The ghost lineage is still missing. The abominable mystery is not fully resolved. And I find that, rather than being a failure, it is a reminder of something important. That the history of life is not a textbook with settled answers. It is an ongoing investigation carried out by thousands of researchers in laboratories and field sites and collections rooms around the world, using techniques that Darwin could not have imagined, finding answers to questions that Darwin formulated with painful precision. The flower changed the world. The revolution it triggered shaped every ecosystem, every food web, every major animal group on land. It built the forests that stabilize climate and generate oxygen. It created the agricultural productivity that feeds civilizations. It produced the ecological conditions in which primate intelligence evolved and from which human cognition eventually emerged. And somewhere, in a warm, shallow lake in the early Cretaceous, it started with one plant. Unnamed, unseen, unbeautiful by any standard we currently apply to flowers, but carrying, in the closed architecture of its carpel, the seed of everything that followed. That, I think, is worth knowing. That is worth the 90 minutes. The world you live in was not always here. It was built slowly, accidentally, without intention or plan, by organisms pursuing nothing more or less than survival and reproduction, over a span of time that makes the entirety of human history look like an afterthought. The next time you see a flower, any flower in a garden, along a road, in a field, I would ask you to try, for just a moment, to see it not as decoration, to see it instead as what it is, a biological instrument of extraordinary sophistication, the product of 135 million years of co-evolution, the visible surface of an invisible revolution that made the world we inhabit. It did not set out to do this. Evolution never sets out to do anything. But it did it anyway, and here we are.